In this short video, I'm going to give you a rundown on meiosis. Here is a cell, and within the cell you can see some chromosomes. There's one and there's two, and these in blue are the homologous pairs. So the first step in meiosis is replication. So first of all, each little chromosome is going to replicate itself, and this is the part where they get thicker and the DNA strands are replicating. After replication, homologous pairs line up. What that means is they kind of find each other and they line up at the middle of the cell. That's called the cell equator. And you can see that cell equator is just the middle of the cell, just like the Earth has an equator. This random lining up in the middle of the cell is what we call independent assortment. To clarify independent assortment, what it means is instead of getting all the paternal chromosomes, so I've put the paternal, or the ones from the father, in blue, and the maternal are the red ones. So they don't line up in order of paternal in blue over here on one side, and then maternal in red on the other side. That would greatly reduce the variety that each gamete would end up with. Instead, homologous pairs line up randomly. So we could get this one on this side and this one on this side, and remembering that humans have got 23, this example only has 2 chromosomes. So if you can imagine, if there's 23 chromosomes, that means independent assortment is responsible for a great deal of variety. The second way that meiosis contributes to variation is through crossing over. So crossing over occurs when homologous chromosomes have paired up at the equator and they exchange segments of genetic material. So those bits that I'm pointing to there is a point at which this exchange may occur. To clarify this, I'm going to separate the little chromosomes to show you what this means. So it means that small segments of the chromosomes will exchange genetic material, and I'm just highlighting the segments that had crossed over, and I'm giving them the opposite colour to show that they have swapped with the opposite chromosome. And recalling that in the second stage of meiosis, each chromatid, and remember a chromatid is half of the chromosome, is going to separate to form a gamete. Now you can see after crossover that each chromatid is no longer identical. This one here has exchanged genetic material with the paternal chromosome, so it is not identical to the chromatid that it had replicated itself from. And so that's another example of how meiosis and crossing over contributes to variation. At the last stage of meiosis 1, the chromosomes are pulled apart. At meiosis 2, the parent cell divides. Chromosomes line up in single file at their equators, at their cell equators. After lining up at the equator, they begin to get pulled apart. Oh, this little section belongs there. So these cells are what are going to become the daughter cells. So that's one, that's the second one, that's the third one, and that's the fourth one. These daughter cells are the gametes, and they have got half the amount of chromosomes that we began with.